Human genetic disorders. What runs in your family? Molecular research has shown a direct link between genotype and phenotype. For example, people of African and Europe ancestry are more likely to have wet earwax, the dominant form. Those of Asia and Native America ancestry most often have dry form, which is recessive. A single DNA-based difference of guanine instead of adenine in the gene for a membrane transport protein causes this protein to produce dry earwax instead of wet earwax. There's a direct connection between molecule and trait, between genotype and phenotype. In other words, there is a molecular basis for genetic disorders. Changes in the gene's DNA sequence can change proteins by altering their amino acid sequences, which may directly affect one's phenotype. Thousands of genetic disorders are caused by changes in individual genes. These changes often affect specific proteins associated with important cellular functions. Sickle cell disease. This disorder is caused by a defective allele for beta globin, one of two polypeptides in hemoglobin, the oxygen-carrying protein in red blood cells. The defective polypeptide makes hemoglobin less soluble, causing hemoglobin molecules to stick together when the blood's oxygen level decreases. The molecules clump into long fibers, forcing cells into a distinctive sickle shape, which gives the disorder its name of sickle cell disease. Sickle-shaped cells are more rigid than normal red blood cells, and they tend to get stuck in the capillaries. If the blood stops moving through the capillaries, damage to cell tissues and even organs can result. As the blood flows, it would be more likely to stick with sickle cells than normal hemoglobin. Cystic fibrosis, CF, is most common among people of European ancestry. Most cases result from the deletion of just 3 bases in the gene for a protein called cystic fibrosis transmembrane conductance regulator, CFTR. As a result, the amino acid phenylalanine is missing from the protein. CFTR normally allows chlorine ions to pass across the cell membranes. The loss of these bases removes a single amino acid, phenylalanine, from CFTR, causing the protein to fold improperly. So the misfolded protein is not used, but recycled. With cell membranes unable to transport chlorine ions, tissues throughout the body malfunction. Children with CF have serious digestive problems and produce thick, heavy mucus that clogs their lungs and breathing passageways. People with one normal copy of the CF allele are unaffected by CF because they can produce enough CFTR to allow their cells to work properly. 2 copies of the defective allele are needed to produce the disorder, which means that cystic fibrosis allele is recessive. Huntington's disease is caused by a dominant allele for protein found in brain cells. The allele for this disease contains a long string of bases in which the codon CAG, coding for the amino acid glutamine, repeats over and over again more than 40 times. Despite the intensive study, the reason why these long strings of glutamine cause disease is still not clear. The symptoms of Huntington's disease, namely mental deterioration and uncontrollable movements, usually do not appear until middle age. The greater the number of codon repeats, the earlier the disease appears and the more severe are its symptoms. Disorders such as sickle cell disease and CF are still common in human populations. In the United States, the sickle cell allele is carried by approximately 1 in 12 of African ancestry, and the CF allele is carried by roughly 1 person in 25 of European ancestry. Why are these alleles still around if they can be fatal for those who carry them? Most African Americans today are descendant from populations that originally lived in West Central Africa, where malaria is common. Malaria is a mosquito borne infection caused by a parasite that lives inside red blood cells. So, malaria, and here is sickle cell allele. Individuals with just one copy of sickle cell allele are generally healthy and are also highly resistant to the parasite, giving them a great advantage against malaria. The upper map shows the part of the world where the malaria is common. The lower map shows regions where people have the sickle cell allele. More than 1,000 years ago, the cities of medieval Europe were ravaged by epidemics of typhoid fever. Typhoid is caused by a bacterium that enters the body through cells in the digestive system. The protein produced by the CF allele helps block the entry of the bacterium. Individuals heterozygous for CF would have had an advantage while living in cities with poor sanitation and polluted water. And because they also carried a normal allele, these individuals would not have suffered from cystic fibrosis. So, what are the effects of errors in meiosis? If non-disjunction occurs during meiosis, gametes with an abnormal number of chromosomes may result, leading to a disorder of chromosome numbers. The most common error in meiosis occurs when homologous chromosomes fail to separate. This mistake is known as non-disjunction, which means not coming apart. Homologous chromosomes fail to separate. These two yellow ones don't separate, they go to one cell during meiosis. Non-disjunction may result in gametes with abnormal number of chromosomes, which can lead to a disorder of chromosome numbers. These two gametes have 3 chromosomes. They're supposed to have 2, and these two have 1, and they're supposed to have 2. If 2 copies of an autosomal chromosome fail to separate during meiosis, an individual may be born with 3 copies of that chromosome. This condition is known as trisomy, meaning 3 bodies. The most common form of trisomy, involving 3 copies of chromosome 21, is Down syndrome, which is often characterized by mild to severe mental challenges and a high frequency of birth defects. So this is the karyotype for Down syndrome, and you'll see chromosome 21 has 3 copies. Non-disjunction of X chromosomes can lead to a disorder known as Turner syndrome. A female with Turner syndrome usually inherits only 1 X chromosome. Women with Turner syndrome are sterile, which means they are unable to reproduce. Their sex organs do not develop at puberty. There's the X, but there's no Y, and there's no second X chromosome. In males, non-disjunction may cause Klinefelter's syndrome, resulting from the inheritance of an X chromosome, which interferes with meiosis and usually prevents these individuals from reproducing. There have been no reported instances of babies born without at least 1 X chromosome. This indicates that the chromosome contains genes that are vital for the survival and development of the embryo.